I usually put them on top of this in the room. Seems perfect height. Doing that today, as a matter of fact.
Since today. All right.
You're purple.
Good morning, everyone. Mr. Dean has an announcement first before we get started.
Okay, IRT decisions are coming up very, very soon, so raise your hand if you're on IRT. Alright, you need to tell one of these people, you need to introduce yourself to one of these people if you're interested in joining the research team, okay? You need to introduce yourself, make sure they know that you're interested in joining. Thank you. That's it.
Today we have DBA presenting on a new type of gene therapy in muscular dystrophy. It's really cool, so DBA.
Good morning.
Good morning.
Great. The title of the article that I read was The Prevention of Muscular Dystrophy in Mice by CRISPR-Cas9 Mediated Editing of Germline DNA. In other words, how do you edit the gene that causes muscular dystrophy and prevent the disease? So how many of you know what muscular dystrophy is? All right, so that means that most of you know that it's a disease that affects the muscles. And it weakens the muscles, and over time, the strength of the muscles decreases. The age of the onset of disease varies, and there are actually 9 different forms of muscular dystrophy. Duchenne and Becker are the 2 most common forms of the disease, and Limb-Girdle is the form of the disease that my mother had. My mother, as a child, walked on her toes, and as she got older, it was noticeable that she was not able to do things that other kids her age were able to do. That said, she had a very productive life. She got married, she gave birth to 2 beautiful daughters. She taught first grade for 30 years. She actually purchased several rental properties and managed them, got her master's, and had a very productive life. She always used a cane as long as I knew her. She had trouble walking up and down steps, trouble getting up from a seated position, and also had trouble lifting heavy things. So things as simple as taking corn pudding and putting it in and out of an oven was something that she would need assistance with. Duchenne muscular dystrophy is the form of muscular dystrophy that was the focus of this particular article. And Duchenne muscular dystrophy is going to affect your shoulders, it's going to affect the top of the trunk, your thigh muscles, your hip muscles, and your calves. And like Limb-Girdle, individuals who are struck with Duchenne muscular dystrophy are going to have trouble walking, running, getting up from a lying down position. They'll experience fatigue very quickly, and their life expectancy is 25 years. Most victims of muscular dystrophy have a very short life expectancy, but with the advances in cardiovascular therapies and respiratory therapies, their life expectancy is increasing. But they're not living longer because of scientific advances to prevent or slow down the progression of the disease. In the 80s, researchers that were funded by the Muscular Dystrophy Association identified the gene dystrophin, which is a protein that is necessary for muscles to do what they're supposed to do. So that's the protein necessary for muscle integrity. What this slide is showing you is basically the dystrophin gene, and this is the dystrophin gene in a normal dystrophin gene. B is showing a dystrophin gene where almost all of the gene is deleted, and that's an indication of Duchenne muscular dystrophy. And C is an indication of Becker, which is a milder form of Duchenne muscular dystrophy. This particular study had two major goals. One, to see how safe and effective the CRISPR-Cas9 gene editing system was in editing genes. CRISPR-Cas9 is a very new gene therapy. And the second is to see whether or not we can use CRISPR-Cas9 to edit the gene and correct the defective dystrophin gene and prevent muscular dystrophy. And basically what the approach was, they were going to take, MDX mice, and those are mice that have a defective, a gene defect at exon 23. So these are basically mice that have muscular dystrophy. And they were going to edit the dystrophin gene in the zygote and implant it in a pregnant mouse, and hopefully give birth to mice without the defect. Simple. So what is CRISPR-Cas9? CRISPR stands for Clustered Regulated Interspaced Short Palindromic Repeat. You may not ever know what that means. Cas9 is a protein that can cut DNA. So think of Cas9 like a scissor. What the CRISPR-Cas9 technique does is it untwists DNA, and then the Cas9 is a protein that actually goes in and snips the DNA. And it's snipped at a location adjacent to the defective sequence. And there are two different methods that they can use to edit or correct the defective gene. One is called, okay, I got it in myself. Yeah, two different methods to correct the defective gene. One is called homologous directed recombination, or HDR, and the other is non-homologous end joining. So just think of the homologous directed correction is one where we actually go in and replace the defective gene, and non-homologous end joining is where we actually snip and delete the defective piece. What this figure is showing is, this is exon 23, where the defect occurs, and this is the gene sequence for dystrophin in a wild type or normal mouse. Underneath it is the gene sequence in the MDX mouse. That's the mouse that has muscular dystrophy. And you can see there's a C to A, the C to T translation, which is the defect. And you have a stop codon that appears where it should not be. What they've done in figure B is they've repeated this defective gene here. Right? And you can see the defect there and the stop codon. and they've matched it up with a sort of like a template of a corrected gene. C is illustrating the HDR method versus the NHEJ method. In this particular study, 7 of the mice underwent the HDR repair, and 4 of the mice underwent the NHEJ repair. So remember, there were 2 objectives of this particular study. One, to make sure that CRISPR-Cas9 did what it was supposed to do, and the second was to see whether or not it was effective in correcting muscular dystrophy. And they tested the CRISPR-Cas9 on wild-type or normal mice, on mice with muscular dystrophy, and also on the corrected mice. And basically what they were trying to do is see if they could edit the genes and see whether or not there were any what they would call off-target effects. So I'm targeting a particular segment of the gene, but will adjacent cells also be affected by this method? And what they learned is that there were minimal off-target effects. In the article, they said that they found the procedure was not promiscuous. I love that. And they also found that CRISPR-Cas9 was effective in correcting 2 to 100% of the gene mutation. They also had to make sure that, they also had to confirm that the dystrophin gene was actually present in the corrected mice. And this is a PCR gel, and they went through normal PCR gel protocol, and they were able to confirm that the dystrophin gene was present in the corrected mice. So this particular diagram is showing the PCR gel. These are showing the levels of correction via the HDR method, and you see it's 41 all the way up to 100% correction. And these are the markers for the corrected mice. So we know that CRISPR-Cas9 is effective to edit genes. All right, now do we know, is it effective to edit the gene that causes muscular dystrophy? So there are 3 different ways to see whether or not muscular dystrophy is present or not. One is to look at the presence of dystrophin in the muscle fibers. And what they did was they looked at the muscles of the heart, diaphragm, ciliates, and the quadriceps in mice, wild-type mice, and as you can see, the green is evidence of a positive presence of the dystrophin gene. And these are the MDX mice. These are the mice that have muscular dystrophy. And you see that there's no green staining, which means that there's little or no presence of dystrophin. And dystrophin is what we need for muscle strength and muscle integrity. These first 2 columns you just saw on the previous slide. And what we've added on here, these are the mice which had 17% correction under the homologous method, 41% correction under the homologous method of correction. And this is for a mouse that had 83% correction using the non-homologous method. And as you can see, as the correction level increases, so does the presence of the dystrophin gene. Okay. Now, this was testing done on mice that were 7 to 9 weeks old. We also did this testing when the mice were 3 weeks old. And they learned that the regeneration of muscle was progressive. So over time, the mice generated more dystrophin, and therefore the muscle integrity improved over time. This is something that they didn't expect to see. So another way to see whether or not muscle integrity is intact is to compare the level of muscle leakage. So if you have muscular dystrophy, there presence of serum creatine ketose, CK. And what this particular slide is showing is if you focus here, these are the wild type or normal mice, and his CK level is 318. This is an MDX corrected mouse, which has the 41% correction, and look at his creatine levels, it's 350. Whereas these guys who have muscular dystrophy, their creatine levels are very, very high. The same with the second litter, where this was the mouse with the 17% correction. His creatine levels are relatively low compared to those with muscular dystrophy. And similarly with the mouse that had the 83% correction, his creatine levels is the lowest out of all of the mice in the litter. A third way is to see whether or not there's muscle strength. So they actually tested the grip strength of the mice. So what they found was that in the normal mouse, the grip strength was about 158, and you can see that the levels are equally high with the mice that had the gene correction. So further evidence that the gene therapy worked. Here's my slide. Okay, I was looking for this one. Okay. All right, so why are these results important? One, they confirm that the CRISPR-Cas9 system is effective to edit genes. So this is another technology that scientists can put and researchers can put in their tool belt for gene correction. The second is that they were able to successfully correct a defective gene that caused muscular dystrophy in mice. And the third reason why it's important is because someone in this room is going to get an idea from this article that may help them discover a therapy or remedy to cure cancer, muscular dystrophy, or some other disease. So what are the next steps? So we need to, you need to, explore the use of CRISPR-Cas9 as an editing system for gene mutations in humans. Also investigate ways in which to improve the HDR and the NHEJ methods, because as you saw from the research, the correction levels range from 2 to 100%. Well, why is there so much variation? Is it because of the tool itself, or is it because of the techniques? And third, investigate the use of the CRISPR-Cas9 gene editing system with other dystrophies and other genetic diseases. Humbling process. The last time I took a science class was in the early 80s. So I had to look up a lot of YouTube videos to look up words, starting with the words in the title of my article. Right? But thank you. So questions? Yes. Do I remember? Yes. Can I explain it? No. I can send you links to some really great videos on how the CRISPR-Cas9 system works and how they target the gene and make sure they get the exact gene sequence that they want.
The surrogate mouse that they implanted what was it? The zygote. Yeah, the zygote and had muscular.
That in some cases it was a wild type mouse. In some cases it was a mouse that had muscular dystrophy.
that like how it had like 2 to 100% correction. You know why it like works so well in some mice and not well at all in others?
So the question is, why did it work so well in some mice and not in others? No, and that's one of the reasons why you need further research in this area. Yes.
Are they planning any other research in the other types of muscular dystrophy?
That wasn't indicated in this article, but the Muscular Dystrophy Association is funding a lot of research on the different forms of muscular dystrophy.
What was the preceding therapy for muscular dystrophy before CRISPR-Cas9, and how is CRISPR-Cas9 improved?
Okay, before this gene therapy, and they've also tried other gene therapy techniques to correct the defect in the gene. There's no medicine that you can take for muscular dystrophy. There's really no treatment for the disease. Okay, so how did they measure the percent correction? So they're basically just looking at a gene sequence, and I'm going from what I interpreted from the article. And if you remember the slide where I showed the 3 different dystrophin genes, and one had the one. Okay, I'll go back to that. So let's say that I wanted to correct all of this, okay? Using my CRISPR-Cas9 method, I might not have corrected all of the defect. So what they're trying to measure, and then that goes back to Akash's question, how come if I might have targeted to correct a particular sequence, all of the sequence, why is it that it was just 2% to 100% correction? So there's some variability there. Okay. Questions? Yes. That's an interesting question. I guess theoretically go back and do more. What they found is that there was, let's say there was only a 2% correction in for that particular mouse. They found that over time there was something that was happening that was causing more muscle to regenerate than the 2%. So they actually measured more correction, or the correction was more effective than the actual amount of the gene that was corrected. So I would think over time, the muscles would get stronger, but I'm not sure whether or not they can go back and then do another round of correction on a mouse in this particular, using this technology. That I know. But you can find it. Yes?
How reversible are the effects of muscular dystrophy? As someone grows older, there's more of an effect on it. There's more damage to muscles, right?
Yes.
So then, how reversible are those effects?
They're, in my experience, and also in my reading, you don't turn back the clock once the muscle starts dying.
So it's more so it stops it.
He's saying
What's
the
that?
treatment. Is there any backtrack? What's the kind of difference?
Like in this.
In the article or
You
just
reverse the effects. What I'm saying is, does CRISPR-Cas9 just stop the muscular dystrophy, or does it reverse it?
They're born.
Oh, the CRISPR-Cas9, that editing protocol, what it's going to do is it's going to correct the defect. All right, there's a defective gene sequence, and it's going to correct that. Then, once the muscle, it's going to correct the defect, and therefore the muscular dystrophy should not develop. That's our goal. you for that.
Next week, Raul was presenting on diabetes and a possible treatment for it. So yeah, come on.
And if any of you want hot chocolate or munchkins, they're back there.
Bagels are fine.
Thank you.
Thank you very much. You're welcome. You're welcome.